Sewers, we take them for granted until things go wrong. These cave scientists risk their lives for their research. And they may even find a cure for cancer. The strange universe inside a common dust ball. But did you know these creatures can harm you? Get ready to visit hidden worlds you've never seen before. Dust. It's everywhere. On the streets, in your house, on your clothes. But there's more to dust than you might think. Scientists are exploring the hidden world within dust, teeming with exotic life. Even in this tidy home on a Washington, D.C. street. It looks peaceful on the outside, but don't be fooled. The dust inside this house conceals all sorts of strange creatures, and it comes from places you wouldn't believe. As head of research and development for Pledge, Armin Klobes knows a thing or two about dust.
We like to think of our houses as castles, but research shows that dust particles floating around outside can easily come into our homes as though there were no doors, windows, or glass. It just comes right in and becomes part of the dust ball under our bed, for example.
Family breathe in every day. In fact, we inhale an average of 10 tablespoons of dust each year, which doesn't cause a problem for most people. What can be a problem is the microscopic plants and animals that lurk in household dust. Molds, mildew, fungi, and bacteria that trigger allergic reactions in millions of people. These hidden house guests are well known to Harvard biologist Harriet Burge.
We share our houses with a whole microscopic population that we can't see. And they live in the dust, they live on the surfaces of our countertops, they live on our skin and fall off of our skin into the dust. And every time you breathe, you're inhaling all those dust particles. They get into your nose and your throat and your lungs.
So what if you want to find out just what and who is living inside your house? In Washington, D.C., a science journalist named Penny Moser decided to do just that. It all began when a common dust ball sparked her curiosity.
10 years ago, I was writing a magazine piece and I was having a terrible time, so I was procrastinating by pulling dust out from behind a bookcase. And all of a sudden, I had it in my hand and I went, "What is this?"
So Moser took her samples to a laboratory, and what she learned is that inside a common dust ball, there's a jungle of hundreds of exotic flora and fauna.
We can see bacteria, we can see mold growing, we can see insect parts, we can see insect feces. And those are the kinds of things that can cause problems for us if we breathe too much of it.
Since Penny Moser did her first exploration, scientists have learned a lot more about dangers concealed in ordinary house dust.
There are two classes of things in dust that can be considered dangerous. The first, and the ones that I'm most interested in, are the allergens. And these include fungal spores that produce allergens, dust mites, cockroaches, cat dander, dog dander, mouse urine, things of that sort.
Armed with this new knowledge, Moser is investigating her home again. This time, she'll use more advanced tools, like this electronic air sampler, to find out what's living and breeding inside her house. Because it's at the whim of moving air, dust is constantly circulating around our homes, seeking undisturbed corners in which to settle. But these dust balls have nowhere to run, nowhere to hide. This is called aggressive sampling. The vacuum pump sucks air through minute holes and deposits airborne particles on the petri dish inside. Once it's cultured at the lab, Moser will have a better idea what she inhales while she's sleeping. In Moser's case, it's scientific curiosity that's driving her to sample the life inside her house. But for 1 out of every 5 Americans, the stakes are much higher. They suffer from allergies, often caused by substances inside their homes. And 13 million Americans suffer from asthma. Deaths from asthma have risen nearly 40% in the last 2 decades, and no one's quite sure why. The answer could lie in state-of-the-art environmental laboratories, like the one which is working with Moser.
Oh, hi, I'm Penny, and I'm here to see Suzanne. Okay. And after we examine it, then we'll go ahead and set up the cultures.
Scientists like Suzanne Blevins are studying the flora and fauna inside typical houses to discover how dust may contribute to asthma and other problems. It's the first step toward understanding how our homes might be making us sick.
See?
One of the first things Blevins looks for is a tiny creature that often causes big problems. A relative of the spider called the dust mite. Scientists believe the dust mite is the #1 culprit in indoor allergies. Dr. Daniel Ein is one of the leading experts on dust mite allergies. After hearing this patient's symptoms, Dr. Ein suspects the cause is dust mites and orders a skin test.
We've known that dust was a problem for allergic people for many, many years, but it was about 10 or 15 years ago that researchers were able to pinpoint the dust mite as the major cause of allergic problems in dust. And with that understanding came a whole different approach to treating dust mite problems and dust allergies.
All right, now we're going to place on the molds.
By introducing possible allergy-causing substances under her skin, including a solution of dust mites, the doctor will be able to see how badly the patient reacts to each one.
Under microscope. It'll make you not want to crawl into bed at night when you think of this being in your bed.
It will take just 15 minutes to find out.
15 minutes? Sure. Okay, I'll get that for you so you can see a picture of that.
At Wright State University in Dayton, Ohio, scientists are trying to determine exactly how dust mites cause allergic reactions in humans. Dr. Larry Arlian figures that if he can understand what makes the mite tick, he may be able to control it.
An important aspect of developing a means of controlling dust mites in homes is understanding the basic biology of the mites. How long do they live? How prolific are they? How fast does a population grow?
Dust mites make their home in your bed, on your mattress and pillow. The average mattress has up to 2 million dust mites. And if you have an old pillow, 25% of its weight could be dust and dust mites.
We have sampled easy chairs in family rooms where we have found about 18,000 mites in the equivalent of about a teaspoon of dust.
The reason they like beds, pillows, and living room couches is simple. That's where they find their food. Dust mites live on dead skin particles that we shed.
It has been estimated that we shed 400,000 particles or skin scales per minute just sitting. So even if we're a couch potato, we produce a great deal of dust. The skin scales then become part of this dust ball that we might find under our bed, and mold starts growing on the skin scales, and then dust mites start feeding on the mold, and so dust goes into dust, and dust begets dust, and the cycle goes on.
So the mites hang out in the same places we do, waiting to feast off our shedding skin.
These mites live in human dwellings because we provide them with the climatic conditions and the food that they require. They all require a fairly high humidity in order to survive. In homes, in order to flourish, the relative humidity must be above 60%. Here's a mating pair, a female dragging a male around. I'm going to move the dish off the
In his search to understand the mite,
here.
Dr. Arlian takes a hands-on approach.
Put my finger under the scope. See them scurrying around on the surface of my skin? There's one crawling over a hair. Letting them crawl on my finger here, in among the hairs on my finger, is somewhat analogous to they're living in the fibers of a carpet. They crawl through the fibers and up and down in their normal daily activity as they reproduce and search for food.
Luckily for humans, dust mites are not parasites. They're not interested in live skin, just dead skin scales that flake off. If they were parasites that fed directly off our bodies, we'd be overwhelmed by their numbers. But what is it about dust mites that an estimated 20 million people are so allergic to? Dr. Arlian has established that dust mite feces contain at least 15 different proteins that cause allergic reactions in humans. To make things more complicated, each allergy sufferer has a unique reaction, like a fingerprint, to different combinations of the 15 proteins.
Some individuals may react to 3 or 4 of that pool of 15, and another individual may react to another set of 5 or 6 of those. And some of them are more potent allergens for specific individuals than others.
So Dr. Arlian and his team are trying to isolate each of the problem proteins. Then they'll create a concentrated extract of each one, which allergists can use to diagnose and treat patients.
Hi.
Hello.
You itching? Let's see what you got. This side has the pollens, and you're reacting to one of the weeds, but that's quite a reaction to the dust mites. That is impressive. Once you've identified that it's the dust mite that's the problem, you can take countermeasures against the dust mite, and hence the understanding of humidity and its role in dust mite procreation, the role of where the dust mites are in your mattresses and so on. This is what they look like. And what they feed off are the skin that we shed from our bodies, and also on certain kinds of molds and other things.
Do they get bigger?
Do they get bigger? No, but they don't get big enough so that it's easy for you to see, no. Their excretions are the size of pollen grains.
Okay.
They get into the air and you breathe them in, and that's what sets up the allergic reactions. They live in your pillows and your mattresses, but there are special covers you can put on your pillows and mattresses that keep the dust mite and their excretions inside. This is a picture of the dust mite. I don't want you to get upset about this, but they're really pretty ugly. They look like they're from outer space.
I'd like to present to you your cultures.
2 weeks after bringing her household samples to the lab, Penny Moser returned to see what's developed.
Lots of critters.
Oh my goodness.
Blevins cultured over 100 different bacteria, fungi, and molds from the swabs and dust balls collected inside Penny's house.
There's not a whole lot of bacteria outside, but there is inside your home.
The amount of bacteria that shares her house was a bit of a shock to Moser.
Your bedroom, 6 million organisms per gram of dust.
Wow.
Yeah, that
is a lot.
That's your
that
bacteria.
Is surprising.
And your
The bacteria are plentiful,
That's what
The fungal is lower.
but it's the molds and fungus that create the most spectacular cultures. By adding a bit of food and water to Moser's dust balls, Blevins was able to cultivate a veritable forest. Her cultures reveal a thriving microscopic world. On Moser's countertops, food mold. In her scrub pad, more bacteria. Beneath her bed, dog dander. And in her toothbrush, yeast. And finally, living in Moser's refrigerator, Blevins discovered the fungus that causes ear infections.
This is something called Aspergillus niger, which is actually a very common organism. It's found over your home. But it is also an agent in something called swimmer's ear.
Oh,
And again, if the ears never really dry out because they're in the water several times a day.
Bacteria are everywhere, but most of the wildlife sharing Moser's home presents no danger to healthy people.
With feet in the refrigerator.
There was one other blessing. The dry winter and Moser's central air conditioning have kept her house relatively free of the dreaded dust mite. But the heat and humidity of the coming summer will likely touch off a dust mite population explosion. And a whole new generation of invisible house guests will settle in.
Go.
In the woods of central Kentucky, a team of scientists is embarking on an unusual research trip. They're heading deep into the past, searching for traces of life before the dinosaurs.
Okay, there's some equipment down. I don't particularly want to carry it down with me if you don't feel the same.
Their route back through time begins with a narrow tube drilled 60 feet into the earth.
Is it wet?
Oh, yeah.
It's a tricky descent, but this team has come prepared for science on the edge.
Okay.
John Holbrook will lead his group of cave geologists through 2 miles of twisting passages before they reach their goal. Like all cave scientists, Holbrook never ceases to be awed by this hidden world.
There's not that many places left on Earth where you can actually go and realize there's never been another human being there before. I've had the privilege of being places inside of caves where I've walked in and looked at the place and realized I'm the first human ever to see this place.
Their mission has brought them to Mammoth Cave, the longest in the world, with over 350 miles of underground passages. For scientists like John Holbrook, caves are unique windows on the past. These passages lead them through layers of ancient ocean beds, laid down like pages in a book millions of years ago. Caves provided sanctuary for some of our earliest ancestors. Native Americans made their way through these caverns 3,000 years ago. And long before that, other life forms evolved in the darkness and adapted over time in the cool, wet cave. Today, these dark passages are home to creatures unlike any on the surface. Another team of scientists, led by Mick Sutton, is searching for these hidden creatures. Today, they are heading into a very different part of Mammoth Cave. To reach this section is a far easier venture. Leaving their ropes behind, they are able to walk in through an unused tourist entrance. But before the day is through, their research will take them to corners of this cave the public never sees. Sutton is a specialist in the unusual adaptations of life underground. Down here, the absence of light and scarcity of food limit the number of species that can exist and encourage some strange relationships.
This one is adapted to life in the cave. It never goes outside the cave, spends its entire life.
Among the few creatures found down here is this sightless beetle. It depends on another insect, the cave cricket.
The reason the crickets wander into the deep cave sites is to lay their eggs. They find a nice sandy patch that's not too dry and not too wet, and that's deep enough, and dig a little hole in the sand and lay an egg in it.
Luckily for the beetle, which survives by eating the cricket eggs. Among the other specially adapted cave species is the sightless fish that swims the pitch-dark waterways, having lost its unnecessary eyes many generations ago. But today, Sutton's team is exploring something few researchers have paid attention to.
A lot of work has been done on the big, obvious wildlife of the place, but very little has been done in the way of looking at the microbiology of caves.
Mammoth Cave is home to bacteria that don't exist anywhere on the surface.
It's a little snug through there.
Sutton and his team believe that these unusual bacteria may provide an important weapon in the fight against cancer. Meanwhile, 10 miles away, the other team has arrived at their research site.
Yes, I did.
A subterranean river called the Logsden.
Okay.
Most caves, including Mammoth, owe their creation to rain and groundwater that seeped through the porous limestone, eventually forming underground rivers like this one. Rainwater contains carbonic acid, like the ordinary carbonated water in this beaker. When carbonic acid comes into contact with limestone, it gently but persistently eats away the soft limestone. But Mammoth Cave contains another kind of rock as well. It's a quartz-like mineral called chert, and when carbonic acid contacts the harder chert, little erosion takes place. It's chert that brings Holbrook and his student, Phil Statler, to the Logsden River.
Oh my goodness, look at this. Yeah, I know.
These branch-like shapes are the object of their search. During the Paleozoic era, about 350 million years ago, this region of North America was covered by a shallow ocean. Here, prehistoric sea creatures burrowed tunnels through the ocean floor in search of food. Then, over time, what had been ocean floor turned into layers of limestone hundreds of feet thick. But the animal burrows filled up with harder chert. And while water washed away the surrounding limestone and created caves, it left the hardened burrows behind. A fossilized record of prehistoric animal life.
Curving like this and like this. Wild.
For Holbrook and his team, these burrows present a nagging mystery.
Most of the burrows we're looking at have a horizontal meandering type behavior. But
They're bigger than almost any other burrows known from the Paleozoic era.
We are seeing a few of these.
We don't know what animal was making these burrows at this point. All we can really assess is what it was doing. And then from there we can start assessing what sorts of animals do that today and sort of figure out at least what kind of an animal we were dealing with back here in the Paleozoic.
This waterfall on the Logsden River may provide some important clues.
We've been told that there's another layer of chert down there underneath the waterfall in a room down there that I've not been to.
I haven't been there.
Tammy hasn't been there, so none of us have seen it. The burrows may be the same down there, they may be different. But we're not going to know till we get down there and have a look.
Their search to understand and identify the ancient burrowing creatures takes Holbrook and Statler over the edge of the falls. It's a dangerous descent. One slip and they'd be swept down a hole at the base of the waterfall.
Okay, no footing. Gotcha. All right. Okay. Yeah, so this is what this stuff looks like on the underside. Well, welcome to the third dimension.
Yeah, really.
Down here, where they've never been before, Holbrook and Statler discover the burrows are several more layers deep.
You've been working on up above.
And then we got the level right around in here that we just seen, where they're kind of coming out of the ceiling and so forth. But it looks like we've still got another level down in here. Now,
seeing the burrows from this new vantage point adds to the researchers' understanding of the animal's behavior. But they still can't identify the creature. So the next step is to take samples of the burrows back to Holbrook's lab. First, Holbrook student Tammy Eifert slices a thin section of burrow, then sands it down and mounts it on a glass slide. Under the microscope, Eifert and Holbrook detect crystallized remnants of feces and seed pods used by the creature to shore up the sides of its burrow, an activity they can compare with certain shrimps and worms today.
This is coinciding with your other pieces of evidence.
It's an important clue in their effort to identify the creature and add one more detail to our understanding of how life evolved.
Filled with something different. We have been able to confirm that there were actually large burrow-making organisms living in Paleozoic time, which before that we've only been able to demonstrate those things existing during the age of dinosaurs, maybe 100 million years later. So we've pushed these things back in time to a whole new era of geologic history and opened the door for the existence of a whole new group of animals that we never even knew existed.
Points where we've got plenty of water input.
Back in the cave, after more than an hour's hike through winding passageways, Mick Sutton's team reaches their destination, Olivia's Dome, a 3-story high chamber deep below ground. Far over on one side, a small trickle of water forms a hidden pool, an unexpected place for a new step forward in cancer research.
It looks like everything is in place here. There's a couple of glass slides and a couple of limestone slides.
These glass microscope slides and small chunks of limestone were placed here 6 months ago by Sutton's colleague, Larry Mallory. A microbiologist and passionate caver, Mallory began this study years ago when he set out to analyze the microscopic life forms in Mammoth Cave. He soon gathered a unique collection of bacterial samples. Then his research took an unexpected turn.
I got a phone call. It was from a Dr. James Bigelow at the Vermont Cancer Center. He said, "You're a cave microbiologist." And I said, "Well, yes, I am." He said, "There aren't very many people like you." And I said, "That's very true." He said, "Well, you know, I'm a caver, and I also am doing work at this cancer center. Have any of your organisms been screened for the production of anti-cancer compounds?" So I said, "No, no one's done that. Don't have. Do you want to?" "Sure, why not?"
Mallory agreed to send samples of his cave bacteria to chemist Jim Bigelow and pharmacologist Miles Hacker. He chose 10 different samples from the cave, each cultured into a bacterial broth and freeze-dried for testing. Bigelow and Hacker then injected each broth into test tubes containing mouse leukemia cells. They hoped that the cave bacteria would attack the cancer cells. While they waited for results, Bigelow again called Mallory.
Then he said, "If 3 of these come back positive, we ought to quit and go into business." At which point we went, "Yeah, right, and laughed. Because these things never work.
Okay, this is going to be D5. Okay.
To assess their results, Bigelow and Hacker used an electrical current to count the number of cancer cells still surviving in the sample. They were stunned by the results.
Looks good. When you test natural products, maybe 1 in 10 or 1 in 20 will have activity. We were shocked that 3 out of the 10 had remarkable activity and 5 out of 10 had actually measurable anti-tumor activity.
Though encouraged, the scientists knew one crucial step lay ahead.
So what I think we'll do is get a...
They would have to return to the cave for more samples, this time to be tested on human cancer cells. Soon, Larry Mallory will collect these samples and begin this second critical round of tests. Then he will know just how useful the bacteria will be in the fight against cancer. Searching caves for a cure for cancer is not as far-fetched as it sounds. Scientists have long tested microorganisms from unlikely locations for effectiveness in healing humans, sometimes with amazing success. In the 1920s, fungus found growing on a moldy petri dish led to the discovery of penicillin, the wonder drug of the 20th century. In their cave bacteria research, Mallory's team was inspired by the hunt for natural cures in other remote settings. Plant life in the Amazon rainforest and bacteria in sponges on the ocean floor are both being intensively studied for their potential in developing new life-saving drugs. Mallory's team believes their work in the cave may hold similar promise. After collecting the new batch of bacteria from the cave, Mallory returns to the lab for more tests. The trick is to identify bacteria that kill only cancer cells, not other healthy cells in the body. For their tests, the scientists have chosen breast cancer, the second leading cause of death from cancer among women. To make the cancer cells stand out, they are dyed pink, and broths of cave bacteria are added. After incubating the cultures, the team checks their results.
Let's get the results.
Sounds good to me.
It's a success.
Wow, they're interesting.
Several wells on the test plate, which had been pink, are now clear, proving that some cave bacteria were able to wipe out the pink cancer cells.
We were amazed that of the first 700 broths that we tested, we found 7 that are actually selective for this one marker in breast cancer.
Hacker confirms their success under the microscope. A sample with normal cancer cells looks like this, the cells clustered into tumors. But what Hacker sees looks very different. The cells exposed to certain cave bacteria have been blown apart. Though it's only a first step, the cave bacteria hold enormous promise in the battle against cancer. With these surprising results, scientists can't help but wonder what other untapped resources might still be lurking deep within Mammoth Cave. What happens when we flush our toilets? Where does it all go? And how can a major rainstorm send things spinning out of control? Welcome to Boston, home of the oldest sewer system in the country. Beneath the city streets, 360 million gallons of raw sewage are flushed through a maze of pipes and tunnels each day. It takes an army of scientists and engineers to make sure it all gets properly treated and pumped safely into the ocean. If they make a wrong move, a flood of infectious waste could escape into houses and streets. And they never know what might get in their way.
The challenge in sewage treatment is to take what everyone flushes down their toilet, which for the most part is about 99% water and 1 part other material, and to make it as pure as possible and to have as little impact on the environment.
It's up to scientist Mike Connor to monitor the impact of sewage on Boston's waters.
Whoa, yeah, it's a little cold for
And
swimming.
he knows how bad things can get. Just 10 years ago, this harbor was one of the most polluted in the nation. because the city's antiquated sewer system was dumping virtually untreated sewage straight into the harbor. The problem was so serious that city beaches were closed more than half of every summer. And the harbor's bottom-feeding flounder were plagued with cancerous tumors. Today, thanks to a new state-of-the-art treatment plant and a corps of specially trained experts, the harbor is on its way to recovery. But there is still one foe that can overwhelm even this sewer SWAT team: rain. This summer, like every summer, a series of storms will dump millions of gallons of rainfall onto the streets of Boston. And this crew, nicknamed the Duck Squad, is heading down to prepare for the oncoming tidal wave of storm water. Entering this underground labyrinth is dangerous business. Layers of airtight clothing protect them from hazardous sewer bacteria. But even more dangerous are the gases generated by decomposing sewage. Special meters measure the levels of hydrogen sulfide and methane, both so volatile that one spark could cause a deadly blast. For safety, they travel in groups of 3. The point man wears a gas detector and oxygen mask. Deadly gases could still be lurking deeper in the tunnel. During the storm, these pipes will fill to bursting point, so the team searches for any breach in the century-old walls. And they never know what else they might find.
We find illegal connections, we find collapses, we find boards, we find shopping carts, we find garbage cans, we find all kinds of stuff. "500 feet down the tunnel, they discover a damaged connection, a potential weakness that must be fixed before the upcoming storm. When the weather hits, it's worse than predicted. But why is rain such a problem for the Boston sewer system? The system works by connecting pipes from every single dwelling to a series of larger and larger pipes, nearly 6,000 miles in all, that eventually carry raw sewage to the city's treatment plant. In most cities, rain isn't a problem, because stormwater is drained separately from sewage. But in Boston's antiquated system, these two networks are one and the same. So whenever it storms, all the rainwater from the streets flows directly into the sewer pipes below, quickly doubling the volume of wastewater that must be disposed of safely.
And when we suddenly get an inch of rain, it changes the way the whole system functions, and we have to respond quickly to be able to handle that pulse input of material into the system.
Caruso, yeah. No, there's no sense in taking a chance.
It's Ed Caruso's job to handle this sudden influx of sewage-contaminated water.
We'll have them cut back a little bit.
It takes just 1 inch of rain to generate 300 million gallons of additional flow. And Caruso knows this downpour will push his team to the limit.
It's constant communication, it's scheduling, it's throwing your hands up and doing the best you can.
And sometimes things can go terribly wrong. A miscalculation can send thousands of gallons of bacteria-infested wastewater into the streets, or even worse, into people's houses. The good news is, this rarely happens.
In order to prevent the flood of rain-swamped sewage from backing up into streets and houses, the system has been designed with built-in relief valves. But the bad news is, to keep houses dry, some of these relief valves spill raw sewage right into Boston's harbor and rivers. In a major rainstorm, Ed Caruso's challenge is to channel the rain into every available pipe and tank in this sewer network. When the system finally does fill up, he has no choice but to release raw sewage into Boston's waterways, a last resort he hopes to avoid. Today's battle will be won or lost here. Boston's sewage treatment plant. On a typical day, 250 million gallons of wastewater are treated with chemicals before being released through these gates into Boston's waters. But with the additional flood of rainwater, the plant will be put to the test.
Yeah, Bruce, flow is increasing about 200 million gallons in the last 45 minutes. Make sure all your units are ready to go.
It's Joe Duplin's job to make sure the treatment plant isn't overwhelmed.
Come on, you just lost that grit chamber? Come on. All right, hold on. Let's see if Chelsea Creek has activated Caruso Pump Station.
As the flow of incoming sewage rises, Joe must get all his equipment online.
When you're going to be pressured all day to keep this plant functioning at its maximum capacity, it kind of sets up situations where you kind of snap at people and everybody gets a little tense. Jimmy, they're 15 million gallons over. Okay. And if we can't get that gate open, we're going to have to think of something else. Yeah.
Meanwhile, under the streets, water levels are getting dangerously high, and the system is being pushed to its maximum. If the storm continues, it's only a matter of minutes before the relief valves must release raw sewage into the harbor.
All right, Bruce, we don't have any other grit chambers available. We're going to need to keep all those grit chambers online. I want to get Bob Steves on the page party and find out.
Back at the treatment plant, the tension's mounting.
Jimmy, I'm getting some false information.
The plant has reached capacity.
Hi, Eddie. How you doing? Good. Eddie,
Duplin calls Ed Caruso to let him know they can't handle any more flow.
Where they're at, cut back 10 million gallons apiece.
At one of the relief stations, Bob Eunice gets orders to ease the pressure in the overflowing pipes. This is one of only a handful of stations equipped for a last-minute partial treatment of the overflow. The sewage is filtered and dosed with chlorine, then dumped into Boston's waters. It's the best they can do in these storm conditions. Then, reluctantly, Ed Caruso takes the final step he was hoping to avoid. He orders his crews to pull the plug at relief valves with no treatment facilities. Releasing raw sewage into the nearby rivers is the only way to protect the surrounding homes from backups in their basements. Two days later, Mike Connor surveys the damage done to the harbor.
The spray is really coming up. You're going to want to be careful.
He and his team drive right to the mouth of several relief pipes that were dumping sewage during the storm.
Real impact of the CSO.
Using a special probe, they monitor the levels of dangerous bacteria in the water.
And turbidity is 7 NTUs, which is actually
The results are encouraging. The combination of treatment and ocean currents have succeeded in keeping the harbor relatively uncontaminated.
This
The water today looks a lot better than yesterday. Yesterday this bottle would have been filled with sort of small particles and kind of a slick, but it looks pretty good today.
Thanks to better treatment facilities and swift action, the tide is beginning to turn in Boston Harbor.
Years ago, you'd go out, you'd see fish dying, you would see floatables in the water, you'd see scum and different plastics. And today, you can go out, and in some places you can see 30 feet right to the bottom. There's a lot of fish coming back. You don't have stuff coming up on beaches, and everything's becoming better for everybody.
The old-growth forest of the Pacific Northwest. These giant trees are more than 500 years old and reach over 25 stories into the air. Now, thanks to new technology and a group of adventurous researchers, the tops of these trees have become a new scientific frontier. For many years, scientists have studied what goes on down here on the forest floor. But what about upstairs, on the roof of the forest? The tops of ancient old-growth trees have always been an inaccessible and unknown world. That is, until now. Ecologist Nalini Nadkarni is a pioneer in the study of forest canopies. She's helping perfect the newest tool in canopy study, a T-shaped construction crane, just like the cranes that dot city skylines. This is just a model. The real crane, nearly 300 feet high, is being put to work in a remote corner of Washington State.
The crane is going to be a fabulous tool for me. Before using rope climbing techniques, I've always been bound to the canopy trunk, to the inner branches in the trunk, and I've been able to put out little catchers and little ways of getting out. But now with the crane, I can get above it. I can get within the canopy crowns. And so to ask questions like I want to ask, which is, what is the impact of canopies on the forest as a whole? Now I can do that.
Nalini Nadkarni began her work on the hidden world of forest canopies in a very different setting. 15 years ago, she designed a special rope slingshot to get her to the top of the Central American rainforest, where she helped open the world's eyes to the incredible diversity of life at the tops of these tropical trees.
Okay.
Thanks to pioneers like Nadkarni, some experts now believe nearly half of all plant and animal species on Earth may be found up here.
How much line do we have down there?
Nadkarni also helped solve a long-standing mystery. Ecologists had known that the soil of the rainforest was too poor in nutrients to account for the lush growth overhead. What Nadkarni proved from her treetop perch is that certain trees draw some of their nutrients directly from moss-like plants that hang from high branches. When these plants, called epiphytes, decompose on the tree branches, they create a layer of nitrogen-rich soil at the tops of the trees, enough soil for the host trees to actually sink roots into.
That to me is very exciting because it shows that there's this interaction going on that is perfectly clear once you climb into the trees, but we never would have known that without climbing into them.
Now, Nadkarni is bringing her expertise and determination to the northern forest canopy. Though lacking the diversity of the tropics, the canopy here does contain treetop lichens, often so abundant they can outweigh the foliage of the tree itself. Nadkarni hopes that the crane will help her learn how these epiphytes draw nutrients from the rain and fog of the Northwest to nourish their host trees. Nadkarni uses the model for a dress rehearsal so she can make the most of her time in the real crane.
In the northwest dawn, Ned Carney prepares to launch her study of the old-growth canopy.
Oh man, this is so great. So what I want to do is get into one of these taller trees. I don't really mind which one. And one that we can get to safely, one with minimal impact on the tree. And see about installing one of these funnels with tubing that we can put a bottle on.
Her goal today is to set out rainwater collection bottles so she can measure moisture levels at the tops of the trees. It requires a risky climb out into the peak of a 225-foot Douglas fir.
Every single movement I make is very calculated, very thought through. Got it. Easy. See, people who work in the canopy move slowly and thoughtfully because they know that every step they take it's important to know where your feet are.
The tricky move pays off. After spraying water on her collector to simulate rain and mist, Nat Carney proves that her simple measuring devices work. Now, she can set up dozens of other rain collectors in treetops throughout the forest. When her results come in, she'll be able to compare moisture and nutrient levels in the canopy with those on the forest floor. A first step in understanding how the epiphytes help these giant trees survive and flourish. But scientists like Ned Carney, who take their research to extremes, are drawn up here by more than the quest for knowledge.
Aesthetically, it's wonderful to be in the canopy. There's some great scenes of beauty here. So, you know, maybe scientists don't always think about that, or they're told that they shouldn't think about that. But to my mind, just the beauty of being in the canopy is something that's important to be in, to study, and to understand.